Succinate dehydrogenase is the only enzyme of TCA cycle which is embedded in the inner mitochondrial membrane. Rest of the three dehydrogenases, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, and malate dehydrogenase, they are present within the mitochondrial matrix. All dehydrogenases, although they all perform the same function, but why there is difference in location? Functions, they all dehydrogenases, as the name suggests, removal of hydrogen. In fact, they remove a reducing equivalent from the substrate molecule, and then they transfer the reducing equivalent to the electron transport chain, and as they travel down the electron transport chain, they release free energy, and that energy is trapped in form of ATP, the so-called oxidative phosphorylation. All of you know very well. And electron transport chain, as shown by these dark black lines, are also embedded in the inner mitochondrial membrane. So it comes from there that succinate dehydrogenase in the inner mitochondrial membrane, it is very close to the electron transport chain, whereas the other enzymes, they are distant from the electron transport chain. So why this difference in location, although their functions are same? The answer lies in the fact that for these three dehydrogenases, the coenzyme used is NAD. And during dehydrogenation reaction, NAD is converted to NADH. Now, NADH, it is a loosely bound coenzyme. It binds to the enzyme apoprotein in a very loose fashion. It can dissociate from the enzyme protein. Apoenzyme say NAD can dissociate, and then it can go to the electron transport chain and donates its reducing equivalent. And here comes the difference. Succinate dehydrogenase does not contain NAD. Rather, it contains FAD, flavin adenine dinucleotide. This is the coenzyme which gets converted to FADH2. Now, this FAD is tightly bound to the enzyme apoprotein. So, therefore, it is also known as prosthetic group. Prosthetic group means so tightly bound with the enzyme apoprotein that it cannot be dissociated without deactivating the enzyme. So FADH2 is tightly bound to the succinate dehydrogenase, shown by this blue thing, blue line. So since it cannot be dissociated from the enzyme protein, consider a situation if this succinate dehydrogenase were to be lying within the mitochondrial matrix. तो जो FADH2 प्रोड्यूस होता वो डिसोसिएट नहीं हो सकता था. It would have to carry it would have to drag the whole bulky enzyme apoprotein along with it to the electron transport chain. Very improbable and difficult situation. So the situation is that succinate dehydrogenase, it lies in close proximity with the electron transport chain, both embedded in the inner mitochondrial membrane, so that FADH2, it can readily donate its reducing equivalents to the electron transport chain. This is unlike NAD, which can dissociate from the enzyme apoprotein. Now, one last thing, both FAD and NAD are called coenzymes. Coenzymes which bind with the organic substances the non-polypeptide organic substances portion of the enzyme. FAD is called prosthetic group because it is tightly bound with the enzyme. NAD is called co-substrate. It can dissociate from the enzyme. So with that additional information, we conclude. Thank you so much.